These are HeLa cells, the famous cell line made in 1951 from Henrietta lacks. We use a lot of them in my lab every week to study viruses. Coming up on TWiV, we'll talk with Professor Philip I. Marcus, the scientist who was the first to clone HeLa cells. This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 197, recorded on May 15th, 2012. Hi, everybody. I'm Vincent Yellow, and you're listening to TWiV, the podcast all about viruses. For today's episode, I traveled to Storrs, Connecticut, back in May of 2012, where I spoke with Professor Philip I. Marcus, a distinguished professor there who spent his career working on viruses and interferon. Professor Marcus began his scientific career in the early 1950s, and if you recall, that was about the time when HeLa cells were developed. And one of his projects was to try and figure out how to clone the cells. That is, how to make populations of cells derived from a single cell. And although we do this all the time nowadays, back then it wasn't so easy. So listen to my conversation with Dr. Marcus as he recalls how he got the idea to clone HeLa cells and how he finally did it. The first person ever to clone HeLa cells. I have your original letter that I first got in February 10th. It's dated February 5th, 2010. That's when you wrote the authors of the textbook of virology. Do you remember that?
Yes. We're going to have a special session on that, or are we going to settle that?
Yeah, we're going to settle.
You were supposed to make the correction.
So we're working on the next edition now.
Okay.
And I'm going to make sure it's fixed. Absolutely. Yeah. And today
I will ask you to think about what I say and recognize Szilard's contribution was not trivial as Puck. The other is plain. There's a plain one in there if you want. That diet ginger ale, one in the back is plain.
Thank you.
Yeah. And when he was being interviewed by Bill Lanouette for this book, which super book.
Genius in the Shadows.
Oh.
This is about Leo Szilard. Is that how you pronounce
Szilard.
it? Szilard. Leo Szilard.
And out of that whole book, there are 2 pages devoted to the biology that I'm going to talk about.
Oh, I see your name and Puck's name. Very good.
Yeah.
All right.
And Lanouette, who got great notices for the book, he really captured the guy, interviewed Puck, and he got one version of cloning. And he interviewed me, and he got another
version. All right.
And so he went back. He went back and forth twice, and checked with a lot of other people. Puck ended up saying that Szilard's contribution was trivial, and I say it was seminal. Fortunately, I think Lynette listened to both, and he came up with this version, which is close to what I'll relate.
All right, ready to go? Okay, well, we've already started.
Okay.
But I'm visiting today in Storrs, Connecticut with Professor Philip Marcus, who is a professor in the Department of Molecular and Cell Biology at the University of Connecticut.
Correct.
We've been planning to have a chat for quite a few years now.
I think so.
So thanks for having me and to talk about the early years maybe of cell culture, we could call it that.
It was the early years, as you can see.
So you before we start talking about science, tell us a little bit of your background. Where are you from originally?
Springfield, Massachusetts.
Springfield, Massachusetts.
Yeah.
Oh, so you're a Northeasterner. all the way.
Absolutely.
And you went to school in Springfield. Where'd you go to college?
I went, well, World War II was on then.
Okay.
And if you volunteered at 17,
Right.
then they would put you in college, the government. And since neither of my parents got beyond the 6th grade, they really didn't know what college was.
All right.
And they had saved up this magnificent fund of $300 to put me through college. So I thought Uncle Sam might be the way to go. And I signed up, and where did they send me? To a place called Storrs, the University of Connecticut, in 1947, I think.
Which is where we are now, right? Although it was probably very different back then.
I'd say about a tenth the size. It's amazing. But we went to school 6 days out of the week,
Right.
what I thought was 24 hours a day. And at the end of that time, I had earned 2 years college credit. Really the way to go. And that launched me into science. My parents thought I should be a violinist, and they wasted money on that. And they finally concluded, after I had caught some spring peepers, frozen them into ice cube trays. And then when my mother served drinks, I'd pop them in, and they would thaw out, and the spring peeper, that was it. She knew I was committed to science. And then after that, during the war, I went to school here, came back, went to UCLA for a semester, then USC, and then to the University of Chicago, where I can pick up the story we're probably most interested in.
Was that for a PhD at the University of Chicago?
No, I got a master's there. I got bachelor's at Southern California.
Okay.
So here I am, a newly minted microbiologist, looking for a job at the University of Chicago. And I get a job in a laboratory that during World War II was devoted to the inactivation of bacteria in droplets of ethylene glycol. And as fate would have it, the person who ran that laboratory during World War II was Ted Puck, my mentor-to-be. I didn't know him then. He wasn't there. He had gone to Colorado. And I was introduced to this sterile room, maybe twice the size of the office here. And I began acting as a microbiologist. They would spray bacteria into this sterile chamber and spray ethylene glycol. And the mixture at the right humidity would inactivate the bacteria. That was the thinking behind it. So here I am in my new job. I walk in early in the morning. I'm a microbiologist, so I'm used to seeing small things. I look into the sterile chamber and I say, "Is that a mouse?" "Yes, it's a mouse in the sterile chamber." So I dutifully reported this. And it was a little disturbing, but nonetheless, there was a mouse in the sterile room. So they cleaned that up. All right. I reported it. I don't think they were too happy about it, but I continued my work. And one of the first things I did there, this is about 1953 now.
Ah, the year I was born.
Was to give a seminar. I thought, oh, that's exciting. You know, you get a job, they pay you, and you give a seminar. So I talked about what was, I thought, very exciting then, this new work of a fellow called Joshua Lederberg, and bacterial conjugation in sex, and all excited. This fellow were like a lead brick. Okay, no more seminars for me.
This was the Department of Microbiology in Chicago or something else?
No, it was a separate unit tied to this ethylene glycol. Okay, then my next project was we've been spraying E. coli into the sterile room, inactivating it, and collecting survivors and growing them up. And we keep doing this over many cycles, and we found that we can convert a gram-negative bacterium into a gram-positive bacterium. And then, okay. And they were preparing a manuscript on this, and they wanted me to see the slides. So I'm looking, they had all the slides lined up, and yes, we started with a nice pink E. coli, and we kept going, then all of a sudden there was a rod, gram-stained rod, and then more and more, and sure enough, there was a population of gram-positive rods. And it didn't take me long to figure out what was going on. What took time was how to tell them what I thought was going on. And as gently as I could, I said, if you heat this preparation out here, you'll find it survives under conditions where this won't be. And I told them these are bacilli and they have spores in them.
Mm-hmm. Right.
Okay, so fine. I made my judgment, and about 3 or 4 weeks later, my contract was terminated. And that turned out to be a turning point in my life. I had a friend at the University of Chicago who knew I was now looking for a position, and she mentioned 2 people that were looking for a research technician, Aaron Novick and Leo Szilard. I had heard of Leo Szilard, the atomic physicist, but they had a laboratory on the other side of campus in the Radiobiology Institute, where Szilard had turned his work from physics and the atomic bomb and the Manhattan Project into biology, sort of a compensation for all the destruction that it caused.
He became part of this phage school eventually, right? With Max. There's a famous phage.
Delbruck.
Max Delbruck, right?
Szilard spent time in Cold Spring Harbor amongst
So Delbruck was also a physicist as well, so the biology began to attract
yes,
physicists, right?
Yes.
Yeah. Okay.
And that was the germ of molecular biology then,
Correct.
actually. So they set up an interview with me, with Aaron Novick and Leo Szilard, meet them at the tennis court. Mm-hmm. Well, all right. And I read later from Lanouette that for some reason Szilard liked to sit by tennis courts.
I see.
And I met both of them. We had a conversation, and Novick's looking at Szilard, and Szilard's looking at Novick, and after about 15 minutes, they hired me. Changed my life entirely. I mean, these people did science at the highest level. So I moved over to them and worked there until Novak went on a sabbatic, and then I worked with Paul Talalay in the Ben May Cancer Lab at the University of Chicago. Again, a fine group of people. They invited my former bosses to come over and discuss their experiment, changing E. coli. No, they didn't bother to attend. But during that period when I was with Szilard, there was a visit from a fellow named Ted Puck. He had established a Department of Biophysics at the University of Colorado Medical School in Denver. And he was looking for a research assistant. And he liked to hire people out of Novik and Szilard's lab, where they were doing mutation of bacteriophage in the chemostat. So I spent a lunch with Ted Puck, and he was telling me what he wanted to accomplish. He'd like to be able to take single mammalian cells and grow them with the ease that the bacteriologists did. You just plate them out, you get colonies, you can then measure the effects of radiation, antibiotics, whatever stress you want. But at the moment, that wasn't possible with mammalian cells.
Can you remind us that this is 1953?
This is 1953, late.
What is the state of mammalian cell culture at this point?
Well, when I heard he was interested, that was pretty heady stuff for new microbiologists, and I said yes, and I started reading up on the subject. So I read about a fellow named Albert Fisher, dating back to 43, and he had the concept that you won't be able to get mammalian cells to grow a single cell because they have evolved with neighbors. Made sense. So it's not going to happen. I ended up in Puck's lab in late summer of '54. And I go to the lab, it's in the basement, his office is upstairs, and he has a postdoctoral person there, Roshan Christensen, and she had capillary tubes all over the laboratory which had single cells in it. And this was based on work that Wilton Earl and Sanford at NCI at the time. They were attempting to clone mammalian cells, and Earl had the notion that the reason single cells won't grow in culture, and they didn't, you needed mass culture, was they were losing some diffusible element, and that he could get around it by diluting the cells, taking them up in capillaries, where you could confirm you had one cell per capillary. And so he did this with hundreds of capillaries. Once the cell was inside, this would be immersed in a petri dish with growth medium. And he was successful to the extent of maybe 1% to 4%.
So how is success measured? By this cell multiplying?
By the cell, a single cell, confirmed microscopically, multiplying to the point where it would grow out of the end of the capillary into the plate, and you now had a clone. So these were mouse L cells, essentially.
So was that the first continuous cell line?
It would be the first continuous line.
Who did that?
Wilton Earl, Stanford, and Likely, I think.
And these are transformed cells, right?
I'm not sure that they were.
But they were...
They were from mouse.
And they were immortal, basically, right?
Yeah,
Okay.
yeah. The problem with that, and that's the lab I walked into when I joined Puck, was 1 to 4% of all the capillaries, and the awkwardness of the capillary. You can't do the kind of experiment you want. So the first thing I tackled was, all right, get rid of the capillaries, and let's go to micro drops. Same concept, small drop, and you could take a petri dish, put a dozen micro drops in it, confirm they have one cell, and you could put them in with different amounts of cells. As long as you put maybe 20 or more into a micro drop.
20 or more cells.
These were HeLa cells.
You switched to HeLas now.
I didn't switch. HeLa cells were there when I came. I think Puck had gotten them from Tuskegee Institute at the time. Okay. So they were there. But they would only grow in mass culture. And even the HeLa cell going down to the capillary did not do well.
So let's explore that briefly. The HeLa cells made by George Guy in 1951, is that correct?
That's about right.
So you could only grow them in
mass
plates.
Culture.
You could trypsinize them and replate them, but you couldn't do single cell.
No, you had to keep the numbers up. And when we went to micro drops, the critical number was about 10 cells
per drop.
If you had that many in, they'd take off and the drop would be confluent. So
if you put one in,
1 to 5, somewhere in there, 1 to 4%, just like Earl's work. Okay. So we weren't ahead. Fortunately for science, this was now late summer 54. There was a visitor to the laboratory by the name of Leo Szilard, the very person whose lab I worked at the University of Chicago. And unbeknownst at the time, he was negotiating a position with Puck in Colorado because his wife, Gertrude Weiss, had a job in a hospital in Colorado. So he came out, and it seemed very natural to take me out to lunch and find out how things are doing. I hadn't been there very long, but did the micro work. And we're at lunch, and I'm telling Szilard, well, you know, it's not going good enough to grow the single cells. That's still the problem. And we chatted about this a while, and then Szilard became very quiet. And I learned at Chicago, when he's quiet, you don't interrupt. So I didn't say a word. And then he finally says, well, you know, if you can grow mass cultures, the secret is not to let the single cell know it's alone. And I'm thinking to myself, oh, atomic physicist, now to the psychobiology of the single cell. Well, it turns out he really meant in the biochemical sense. And we spent the rest of the lunch working out a system where you could put a single cell in the environment of a mass of cells so that whatever this diffusible substance or substances or factors was, there'd be plenty of them. And I rue to this day, we grew out a schematic on a paper napkin. I don't have that. And we worked out a system that should fit Szilard's requirements not to let the single cell know it was alone. And I'm all excited about this. I'd been there maybe with Puck only 2 or 3 months. And lunch was over, and I said, I'll go to see Puck and tell him about this exciting So I go in and I see Puck, and I tell him about this. And he's quiet. And I said to myself, gee, maybe all great scientists are quiet, so you have to be quiet and listen to them. And then finally he spoke, and he said, no, that's not going to work. Forget about it. And I was absolutely crushed. And I did forget about it. For about 20 to 30 seconds, that's how long it took me to leave his office. I got outside and I said, no, this is too good. I said, I'll try it. If it doesn't work, we'll know. If it works, I'll worry about it. So I went to see the machinist in the biophysics department, a fellow named Bob Edgerton, and I showed him this schematic. Could he make these little plastic platforms? And he cut some out of plastic, and I sterilized them with UV, and I started an experiment on Friday. I put down a mass culture in a small petri dish. I put down this platform, which in practical terms was about 200 cell widths or diameters.
What does this look like? It's just a round cylinder sitting on the bottom?
No, I have a picture of
Okay.
it. You'll see later. It's sort of flat, but it's raised above the monolayer. And on that, I separately put microscope slides that had been cut, glass slides, about so big, and inoculated mini drops again with one cell, or no cell, taking advantage of the Poisson distribution. Many of them were zeros, but I could confirm microscopically they were one. They would settle down and attach in a different dish.
These were HeLa cells?
These are the HeLa cells. Once they're attached, they're very stable. Then I would take the cover slips, place them on this plastic platform, 2 of them, then submerge them in the medium where the mass culture was.
And the mass culture is HeLa cells also?
Yes. Put the cover on, put it away. I did this on a Friday. I came in Saturday. Every place I had put 1 cell, there were 2. Every single cell had doubled. Oh, that's nice. And I came in on Sunday, and every place that had 2 cells now had 4 cells. I said, that's even nicer. And I said, it's working. So on Sunday, I called up Szilard, and he came down to the lab with his wife, Gertrude. And I showed him this, and he gets this big grin on his face. And he's just a big person. He was satisfied right there. It worked, okay? That's it. And then he turned to me, he says, well, you have to call Puck. And I said, well, I wasn't authorized to do this experiment. So he said, no, you have to call Puck. Of course, he was right. I called Puck. He came down to the lab on Sunday. And I told him the setup, and he looked through the microscope. He saw these 4-cell clones. And he didn't say a thing. He just disappeared. And the one thing I cannot remember is how long I didn't see him for, okay? It had to be days. His office was upstairs, the lab was downstairs. He was not a lab person. I think once in all the time I was there, I asked for help. And so.
You were a research assistant at this time,
I was a graduate student
now you're
at the time.
A graduate student, okay.
Yep. Going from master's to PhD in microbiology with also in biophysics. So I think it was a few days or a week. Then he came down to the laboratory as if nothing had happened. And he says, what we have to do is irradiate the bottom layer so that it will be biologically functional but sterile. Mm-hmm. And then it can't contaminate the single cells. Not that it ever did in any of the experiments I did. That's why I was making the point. The platform was in cell diameters, 200 cell diameters. So if a cell came off in mitosis, they go down by gravity. They're not going to go up 200 cells.
So it's a raised platform.
It's a raised platform.
Okay. Did you ever do controls where you just put a platform in without any cells?
I didn't, but I saw no evidence of gaining any colonies. I would verify the microdrops.
Yeah. You start with one, the next day there'd be two, and then four.
Yeah,
okay.
And I didn't pick up one later on.
Yeah.
And the physics, if I can use the term, was against it. Mm-hmm. So, great. And we went ahead and used the X-ray machine that Puck had bought for the department 2 or 3 years earlier, and had never been used. We began to X-ray cells to sterilize them. And it worked. And I'll show you some pictures later of the X-ray giant cells. They produce large, they would undergo cell division, but not nuclear, nuclear division, but not cell division. So the cells got bigger and bigger, but they were sterile.
And the idea is they're producing
whatever the nutrients were that the mass culture was,
yeah.
There's every reason to feel that they were doing the same. It would take, after a dose of irradiation, you could plate them and they get larger and larger for about 8 or 9 days before they disintegrate. So, at about this time, we used the X-rayed feeder layers and the platform that Szilard and I had suggested, and that was the first publication. That's at 1955.
Who was on that paper? You?
Puck and me.
You and Puck, okay. Not Szilard.
Not Szilard. You have to remember, I'm a graduate student now, maybe only 4 or 5 months. Not quite used to how these things work. Besides, Puck had put a footnote in that paper stating Szilard's contribution. I'll discuss that later when we go over the slides, because it's a critical statement. But at the time, if that's the way they do it, that's the way they do it. And so that paper, let's see, that's probably a good place to take a break, because that paper came out. That was the paper that introduced the world to feeder cells. It was feeder cells that Szilard had actually recommended. And Puck had added the element of X-raying them so they wouldn't be able to divide. And together, that constituted the first paper.
I'm looking it up at this moment.
Okay.
See if I can find it.
I have given away all my reprints. I should have it right here to show you. I have it on this.
Okay.
And you will see it in the slide. It's a middle picture.
Okay, so that's a diagram of this platform.
That is the
apparatus.
diagram, and it was drawn by yours truly. And as I look back on it, my gosh, that's a lousy job. One part of the petri dish is hanging over further than the other, and it's a little crooked, but nonetheless,
Well, you had to draw
that's my
with
figure.
rulers and a pencil, right?
Yeah.
That's fine.
Yeah.
I see. So then the cells are growing on this platform. That's great. And the paper described this
This
system.
procedure,
Okay, using HeLa cells, right?
using HeLa cells. Now, we can go to, you want to go to the figure 6 in that?
Sure. Slide number 6.
It's the last page. In figure 6 is the footnote that was in the paper, that 1955 paper. It says, in our earliest experiments, test cells and feeder cells were placed in the same layer. We wish to thank Dr. Leo Szilard, who suggested the more advantageous geometric arrangement in which the test cells were placed on top of the layer of the feeder cell. That statement is not accurate. This was the first experiment we did. That was followed by the fact that we could plate the single HeLa cells in a layer of giant cells. There was enough room, they would settle in, and they would grow, being fed by the giant cell. You didn't need the platform. But the first experiment was the platform as suggested by Szilard, and this statement is inaccurate, leading one to think that we had seeded the cells.
So you didn't do that yourself. You never put the feeder and the test cells in
Later.
Later on, yeah.
Later, we used that procedure.
And before you came to the lab, this hadn't been done?
Not prior to Szilard, none of this work had been done. And in truth, I was the only one working on it. And I was in the lab, and Buck never came down to the lab. And it was at that point that I did cloning and developed these cloning cylinders. I have some out there, you can see them. And cloned the original HeLa when it was plated. You can see here on this slide was quite heterogeneous. Page 5.
So these are clones of HeLa cells growing on a plate.
That's correct. Because finally the media got better and we didn't even need
The feeder layer
the feeder.
any longer. How long did
But
that
you
take?
can, maybe a year or so. But feeder cells are still used to grow stem cells and fastidious cells. When you don't know why they're not growing, you stick in a feeder. Well, here is the wild type, uncloned HeLa. And then I picked the clone from them, and you can see the uniformity picked up, and I'll show you.
So that is the first time that HeLas were cloned?
Yes, using the cloning cylinders.
So you would take a glass cylinder and place it on these colonies and remove some cells.
Well, it was stainless steel, but...
Steel, yes.
Yeah.
We used to use glass.
Yeah.
And you would put a little trypsin in to remove the cells?
You put the silicone on the
Right.
bottom,
To seal it,
and
yeah.
you could press it right over the colony.
Right.
It would stick, remove the medium, then add trypsin, and let them come up and move them over.
And these above are photographs of the colonies, the individual colonies of HeLa
Those
cells?
happen not to be. These are different clones that came from the wild type. The ones in phase contract
Are not HeLa cells?
are probably not HeLa cells. My associate Margaret Sigelak reminds me they're probably wish cells.
So this cloning, which was done with, it was the first time a mammalian cell had been cloned in culture. So it was done first for HeLa before the mouse L929 cell that you mentioned before. Is that right?
No, the 929 cells were cloned in capillaries.
Very inefficient, right?
Yes.
Okay.
But they predated this.
Got that. And then eventually you
in
could
fact,
use your...
we have that picture here.
That's a picture of a capillary,
Yeah.
right? That would be interesting. And you can see the cells growing out
They
of the end
finally
of the
grow
capillary.
out. Here it is, single cell,
I
and they
see.
they take off.
So I can put these in in our video, right?
They're
That'll
in
be
there.
great.
That is a duplicate of what. The only thing I left out is a cloning soldier. You take one back with you.
So the eventually you adapted the HeLa procedure to these L cells or any other cells to get clones. Right?
Oh, once the HeLa,
Then everyone else followed, right?
yeah.
And today, cloning cells is very important. If you want to put a gene in a cell, you have to pick a clone with just that gene. So it must remind you all the time of the
Yes,
early
yes.
work that you did, right?
Yeah.
Is this a photo of you here in the second slide?
That's when I was at the University of Chicago working in Novick and Zilar's lab.
Okay.
So that takes us back to 53.
So that year,
Then I joined Puck.
Puck, right?
This is from Harris's book.
And here's a photograph of Szilard
And his friend. When I worked in Zilar's lab, it was a little unusual, a primitive one might say. There was one phone. It was on the wall. And when Szilard would come back from his trips around the world, fertilizing ideas to different scientists, he'd come back and he'd make all his phone calls on that one phone. I'm working in the laboratory, and one day I hear him speaking to the University of Chicago operator. Would you please get me Professor Albert Einstein at Princeton? So I did hear one part of the conversation. It was in German. My German wasn't that good, but I ended up at Einstein College of Medicine.
So the science was very different back then. It was much less formal than it is now, right? The phone in the lab.
Yes, that, plus the fact, if you had a respectable proposal, you could get it funded. And papers were written then, they would open up with, so-and-so showed that this, and then your work would follow. You show me a paper today that says so-and-so did this. It's changed.
Why do you think that's so?
Because of the scarcity of money, the tight budgets.
Mm-hmm. Yeah, it's much more competitive now.
Extremely so.
So you worked how long in Puck's lab?
I worked from 54 to 60. Then 60, I went to Albert Einstein College of Medicine.
And that was as a faculty member there?
As an assistant professor, and Szilard had sent a reference, and he wanted to know why they didn't hire me as an associate professor. That's Szilard.
And tell us who was at Einstein at the time that you remember? Because I remember a lot of well-known virologists. Bernie Fields was at Einstein, right?
Bernie Fields.
Was he there at the time you went?
I don't think so. Yaklik was there.
Bill Yaklik. Now I know Don Summers and Ellie Ehrenfeld were there.
They were there.
The same time that you were there?
Yes.
And the DNA replication guy.
Oh, yes. Oh, right.
He may still be at Einstein, actually, right?
Is that right?
I forgot his name.
I know his name.
You know who I'm talking about, right?
Yeah, Yeah,
And
great guy.
And I'm terrible. I can't remember his name. I don't have internet, or I would look it up.
I haven't thought of his name in years and years.
Yeah. No, I think he might be at Sloan Kettering now. No, probably still at Einstein, but still doing DNA replication.
No, the fellow, Jerry
Hurwitz. Here we
was
go.
there.
So he was there.
And
Okay.
Philip White. I mean, they weren't virologists, but they were top-notch
Okay.
biochemists.
And what did you work on in your first lab?
I worked on the phenomenon, the first demonstration of the dynamic incorporation of a viral synthesized molecule in the cell, its transport to the cell surface, and its movement around the cell. You don't know about that paper. It came out in 1962. You were only so high then.
I was 9 years old at the
Yeah.
time. But this was influenza virus, it sounds like?
It was NDV and HeLa cells. I can show you some pictures later. But it turns out to be a classic because it showed how the hemagglutinin molecules first appeared at the polar ends of a cell, if it was bipolar. Did I have one here? No. And if it was tripolar, the red cells would stick on the tips. And then they would move in centripetally until the whole cell was hemabsorption positive. And after working that out, I spent a year with Delbecco at La Jolla and came back to Einstein, and we were in the midst of a rubella epidemic. And I had a good colleague in pediatrics, Dave Carver, and he says, you have to develop an assay for rubella virus. They don't have one. This is not a CP. It doesn't destroy cells, so you have to do something indirect. For some reason, I said, okay, let's see if NDV interferes, let's see if rubella virus interferes with the replication of NDV. So we infect cells, viro cells, with rubella virus. You go back the next day, they're fine. 2 days, 3 days, they're fine. No CPE. But if you challenge those cells with NDV, the high multiplicity, they're all infected, they were refractory. They would not absorb red blood cell. So we turned that into what is called a hemabsorption negative plaque assay. And there are several viruses that are non-cytopathic that you can get plaques with that way. And someone went a little further, this is years ago, and said, well, using this, we call it intrinsic interference. The only cell that was blocked by NDV was the cell that had rubella. Right next to it, no rubella, you got NDV to go. This fellow let the cells go, and they killed all the cells around the original hemabsorption negative plaques, and you had a colony. You could tell it that way.
So you were a few years, I wanted to bring up Delbecco, who, as you know, just passed away recently.
Yes.
So
Yeah.
you had spent
I
some
spent
time,
a year there. Great
and
place.
he embraced cell culture very early on.
I would say he probably was a pioneer in that sense with respect to virologists. I remember Puck coming back from a meeting at Caltech, and he looked through the microscope and he says, that's a metaphase cell. He got that from Delbecco.
I see.
Yeah. That was a heady time there because Puck had Sir McFarland Burnett before he was Sir, and I met Jim Watson and Pauling, Delbecco, just stellar people
Sure.
visited.
So Delbecco developed the plaque assay for animal viruses, of course,
Yes,
in the early 50s.
yes.
And then I know that Howard Temin went to work with him and then used the cells to demonstrate transformation by RNA tumor viruses. So
When Delbecco was plaquing polio on HeLa cells, I think, yes. And he would use neutral red to contrast it. And he decided, well, to get more contrast, he painted the laboratory green. So when he held them up, the red against the green gave you more contrast.
nice. Very good. That was brilliant.
Yeah.
That must have been some time in the 50s, in the early days of cell culture, combining it with virology. But of course, in Puck's lab, you weren't interested in virology. You were interested in cell culture.
It's
right?
interesting. His work, I learned after the fact. He was usually didn't discuss these things, was supported by the
The National Foundation.
National Foundation for Polio.
Hmm.
And I was the first one who used the cloning and the virus combined. I had learned my plaquing from Seymour Levine. He was a postdoc at the time. And another interesting tidbit, I plaqued WEE on chicken cells.
Western equine encephalitis.
Yes.
Okay.
Which, as you know, is infectious for horses and people. Well, they didn't have a vaccine at the time for people. So guess what? We got the horse vaccine.
Oh, that's something you couldn't do today, right?
I don't think you could do it today, not knowingly anyway.
Yeah.
And so I get these beautiful plaques of WEE on chick cells, my first plaque experience. And I got busy with another experiment. I had added liquid neutral red to help visualize it. So I put the plaque plates back into the incubator thinking, oh, I want to see if they keep growing. I'd forgotten about it, came back 2 days later, took the plate out, and it had a very unusual appearance. I can show you some picture. The original plaques were there. They had not increased in size. They were surrounded by neutral red stained cells. But between the plaque areas, all the cells were destroyed.
Yeah. Interesting.
After the fact, I realized I was working with interferon. I had taken this up to Puck, and I said, look at this strain. He says, oh, we see this in phage all the time. That was that. But I never forgot that. I even took a picture of that plate. And in 1962, when I was talking at Cold Spring Harbor, their symposia on this the mobility of the hemagglutinin on the cell surface. I'm sitting next to a fellow who was going to give the lecture after me up front. And we start chatting, and he's talking about this interferon. What's the interferon? I never heard of interferon. It was Alec Isaacs, co-discoverer of interferon. And I think it wasn't many years later that I introduced that in the Cold Spring Harbor course I taught with several people. Gordon Sato. Who's this fellow from Rockefeller? Richard Franklin. You must know his work.
Well, he was David Baltimore's PhD advisor at Rockefeller.
Okay.
And he worked on Mengo virus. And polio eventually, and that's where David
Yeah, we gave the
started working on RNA polymerases. So I found the reference to your paper, Puck and Marcus, 1955, a rapid method for viable cell titration and clone production with HeLa cells in tissue culture, the use of x-irradiated cells to supply conditioning factors.
Exactly.
So I work with HeLa cells all the time to study viruses, and we use a clone called S3.
Yes.
So were these clones made using your technique?
They were not only made using my technique, they were made by me.
Really?
Yes. So I one day cloned 3 clones from HeLa, from the wild type. 1, 2, and 3. And Puck added the letter S, which stands for Florence Sabin, whose the laboratory was named after. Quite a renowned.
No relation to Albert Sabin.
No relation to Albert Sabin. So S1, S2, and S3. And you know S1 is still around?
I didn't know that, no.
S3 is the most popular. Right. S2 you don't hear about because I caught a cold one day and I'm working with transferring S2 and it disappeared.
I see.
That's the story of S2. S1 turned out to be deficient in the amount of inositol it put out and required. And so if you plated it, the plating efficiency was maybe only 10 or 20%, as opposed to S3, which was 100%. So that's why you don't hear much about S1.
Well, we use S3. I have used them since 1979. And I never knew who cloned them, and now it's really nice to know that you did that years ago. And you probably know that many, many people use these cells in many different laboratories.
Well, I gather if you counted them all up.
That's quite remarkable. I like
it. It was a remarkable cell in that it grew like wildfire. You plate clone, if the cell happened to be located near the edge of the plate, it would form a colony and it'd start completed on the side of the plate.
Right. Do we understand why HeLa cells grow so well?
Probably because its chromosome composition is so complex and so multi. It's just pouring out everything.
Because no other cell line approaches it, really, right? Or is that not true?
I'm not sure that that's the case. Vero cells do very well. We did the cloning with influenza virus that John Gogiri did using Vero cells. And we hadn't been using them for cloning, and the plating efficiency was low, and I told him just keep rapidly passing them, and you select for it, and now we have a line that clones well.
So I want to ask you one more thing, and
Surely.
You can, if you want, we can not talk about this, and we can delete it. But as you know, Puck wrote an article saying that your version of this story isn't correct. So would you rather not discuss it, or?
Oh, I don't. He wrote the article. He was interviewed by Bill Lanouette.
This is for this book on Leos
for that
Szilard.
book.
Bill Lanouette, Genius in the Shadows.
Right. And out of, Lanouette tells me, out of all of this book, the file on these 2 pages is thicker than any other file he's got, trying to get the story straight. And the reason I don't hesitate to comment on it is it took me 50 plus years to figure out what footnote 6 meant. What it meant was we did this before by coplating And then Szilard offered this platform. That isn't the way it happened. And I know because I did both.
Right.
I was the only one working in the lab on that. And there's another clue to why his version isn't correct. He wrote a small monograph called The Mammalian Cell as Microorganism. When I was there, he mentioned that he was writing a book and he couldn't think of a title. So I was reading something about clones from McFarlane Burnett's book. And so I gave him this title. That's fine, okay. And Puck wrote the book, which is unique in there's no attribution in it to any of the figures that are there. And in my naivete as a graduate student, I'd find something going on in the lab that was fascinating. We had an incubator with a iron grid where you put the bottles or the glass plates. And I'm sorry I don't have his book here. And when I looked at the cells plated, they followed the iron grid pattern.
Right.
Showing how sensitive they were to heat. In between where it was air, they weren't growing. And I took this up and it incorporated, and most all of the early material I had supplied. But the thing that amazes me about that book, and I've been over it twice, it does not quote the original 55 paper in the opening chapters. It says selected references. And the selection was against the original paper.
I see.
Now, why would someone do that?
We will never know.
We will never know, but footnote 6 is there.
Right, right. And Dr. Puck has passed away,
Yep.
right? He seemed to be offended by Szilard interfering in his laboratory.
Yes. And I am so sorry about that. As I said, I did not know at the time they were negotiating. However, Puck accused Szilard of being an interloper, interacting with his students. I had worked with Szilard. He was visiting. He was a few doors down from me. Nothing could have been more natural to have that meeting. Now, over the years, I don't have to tell you, someone comes up to you as a mentor and presents you with a solution to a problem that you've spent a lot of time on. How big a person you are determines how you receive that information.
Of course.
And to Lanowet's credit, apparently in a letter that Puck wrote to Szilard saying he's sorry there wouldn't be a position there. I can probably find it later. It's quoted here. But your brain is so much stronger than mine that I don't think we would work together. And that's in the book.
It seems to me that you would want someone as brilliant as Szilard to be helping you, right?
I would say every time there's an opening in the department, I said, go outside and get the best person you can.
But Szilard had no ulterior motive for helping you, other than he
Absolutely
was
not.
interested, right?
No, he got his delight came from seeing it work, and that's it.
Right.
And he did this throughout the world. He would visit Monod's laboratory. He was behind some of the basic thinking there.
Truly altruistic, right?
Uh,
Wonderful story.
A great person.
I know that we have the attribution of cloning HeLa cells wrong in our textbook, so we will fix that in the next edition.
I think you're recorded in saying that.
It's fine, no problem. Because I'm one of the co-authors, as you know, and I'll make sure it's fixed.
We use your textbook, as you know. I send you whatever. It's sitting up there
I
next
see it
to.
in several editions.
Yeah.
And you teach a virology course here to undergraduates, is that right?
Yep.
Do you talk about these early days of cloning?
I do.
Do they appreciate it, do you think?
I actually ask them if they do, and some of them do. It's a little heady for them, but lately I have been speaking to a colleague's class in ethics, and so I've gotten together these slides, and in the last day of the semester, I spoke to his class and covered much of the material we're discussing now. And when I was finished, the class applauded. That's a rare event in most classes. So I think they got the picture, because it's become clearer and clearer to me as I think back on it. And the evidence, no quotation of the original paper in the book, the footnote, which is not accurate, trivial contribution, there it is in the first paper, the platform.
So you said you record your lectures?
Yes.
Are they online, or are they just for
No,
you?
I have them all on disc. Margaret Sekelica sends them over to the Yukon Husky site. Everyone signed up for the course has access to them, and she does slides. Every slide I
Wonderful.
go up.
Great resource.
It is. They like it. They love it. And it's not satisfying. Now they want it streaming, so when they're walking along, they can listen.
Of course.
Of course.
It's technology.
But you've come to the ultimate of technology.
Well, I put my lectures online, and anyone can access them. This semester, I just finished teaching my class. We had 14,000 people subscribing to my course, which I think is amazing.
Fantastic. I thought I had set aside. Well, I'll find it. I have one of the lectures that describes the acquisition of new viral molecules on the cell surface. I meant to give that to you.
Well, I'm going to thank you for taking time to tell this story today. I appreciate it. And I think it's good for all of our listeners to hear it from someone who was there, because there aren't many people who lived through the very early days of cell culture and virology, so it's good to hear this story.
It was rather primitive. This picture you see here, right. These are the glass pieces. There were 5 colonies placed, single cells placed here, 5 times more placed on this one, so there were 25. And this is a picture of one of the clones. And we didn't have any equipment to take pictures of clones, apparently. So Puck goes and finds this commercial ad, and this fellow comes with a bellows that long, and he sets it up on a tripod. And the bellows is way out here, and he's got his film there, and this is the result of it.
Wow. Yeah,
So,
I was going to say you probably couldn't photograph the results very readily. But nowadays, of course, it's easy. How things change, right? All right, thanks so much.
So, we take a break for lunch, and I can show you some real clones.
Okay.
I hope you enjoyed my conversation with Professor Marcus. As you can see, he is a pioneer, and I think it's important that we talk with the pioneers in science and keep their discussions on record for everyone to hear. And my conversation with Dr. Marcus will always be available free of charge for everyone to see and listen to on TWiV. You can find TWiV at twiv.tv and also on iTunes. If you do like TWiV, please go over to iTunes and subscribe and leave a rating or a comment. That helps us to stay visible at the iTunes store so that more people can learn about the wonderful science that we discuss. As always, if you have any questions or comments, please send them to twiv at twiv.tv. I'm Vincent Racaniello, and you can find me at my blog, virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.